Coffee cultivation is one of the most important agricultural practices worldwide, with an annual production of 10 million tons with a value of $70 billion. In Guatemala, a variety of Coffea arabica species are cultivated, and C. robusta is the most important one as it has shown tolerance to pests and diseases. When researching and interviewing farmers from our country, Guatemala, we found that rust continues to be a problem for their crops. Currently, the country has lost $543 million in 5 years due to coffee rust. Coffee rust, Emilia bastatrix, is a phytopathogenic fungus that affects the productivity of crops by weakening the coffee plants and causing death to their branches. This disease represents one of the major causes of production losses, which reach up to $2 billion. In order to stop these losses, we are finding a viable solution in order to help farmers and the country's economy.
Helping people in our country is a prime priority for us. A big part of our country's population works in agriculture. Therefore, they are affected by the loss of coffee crops due to fungi. In order to solve this problem, we develop a spray that induces the production of antifungal proteins by the coffee plants. This will make the coffee plant resistant to the coffee rust and reduce the coffee losses. By applying our product in crops, the profits of farmers will increase, and we will give a hand to our country's economy.
To establish the final idea of our product, we had to develop several proposals and design different plasmids. However, thanks to the feedback of our advisors, we found what we consider to be a viable proposal to solve the problem of coffee rust in our country. The following is an explanation of our project.
We propose a Gibson assembly method for the construction of our vector. This technique was chosen because the construction of our plasmid requires the cloning of multiple sequences for both antifungal proteins. These sequences can be obtained via a PCR reaction with the properly constructed Gibson assembly primers, which will allow us to incorporate the overlapping ends to each fragment for the insertion of the coding sequences for both proteins into the plasmid. We kept in mind that this protein expression cassette was added to the multiple cloning site of the mentioned plasmid in the EcoR1 restriction site. Therefore, the primers for both ends of the protein expression sequence were constructed based on the left and right arm of the digested vector, contemplating the overlapping region sequences in order to allow the annealing of the inserts into the plasmid. Both the expression cassette fragments and the digested vector will go through the same master mix reaction in order to generate the modified pCambia2301 plasmid that will allow the proper expression of the antifungal proteins in the coffee trees. To make identical copies of the designed plasmid, we will use E. coli since it grows quickly and easily in the majority of media, is easy to genetically engineer, and it has great plasmid stability. The plasmid transformation will be made by subjecting these bacteria to electroporation. After the plasmid introduction in E. coli, a selection must be made to select which cells were effectively transformed. For this, we will use the dominant selection marker kanamycin, which inhibits protein synthesis, as our primary selection method, and beta-galactosidase screening as the secondary selection method. The antibiotic resistance is used to indicate that the plasmid is in the bacteria, and the second method to indicate that the desired sequences were introduced in the LAC-C operand of the original plasmid, so white colonies are expected instead of blue ones, which do not contain the desired sequences. We need to reproduce the plasmid by growing E. coli in a reactor and supplying all the required substrates in proper concentrations to promote growth and avoid inhibition. The bacteria will enter the growth phase at 30 degrees Celsius, and when a proper cell density is reached, the temperature will be increased to 42 degrees Celsius for 10 hours to induce the plasmid DNA production and expression. To recover the plasmids, the cell biomass will be separated by centrifugation. An alkaline lysis with SDS and sodium hydroxide will be used to separate the plasmids from the chromosomal DNA and other cell debris. Carbon dots are an essential part of our project. We want to use them as a tool to deliver the plasmid. They have a very special characteristic for us, a positive charge, that will let us attach the plasmid with a negative charge to them by electrostatic interaction. To enter the plant, the carbon dot is recognized as a carbon source, so the stomata will open and let the carbon dot with the plasmid in. Then the plant cells are going to do endocytosis because the carbon dot bind with the plasmid will have switterionic characteristics. Inside the cell, the carbon dot and the plasmid would separate. The plasmid would enter the nucleus and make the transformation to induce the antifungal proteins production, and the carbon dots would be excreted or assimilated by the plant. To promote a circular economy, as carbon source we want to use the coffee beans that are wasted because they don't meet quality standards. And after the synthesis of the neutral carbon dots, they will be added as the surface passivation agent to give them a positive charge. To apply the product, a solution with the plasmids, tea buffer, and carbon dots will be prepared and will be sprayed in the morning and in the afternoon for 5 days at 10 centimeters away from the plant until the leaves are wet. And controls will be prepared by excluding carbon dots in the spray, excluding the plasmids, and excluding both to confirm that the carbon dots can be used as a transfection method to introduce DNA in the coffee plants that calls for the expression of antifungal proteins that confer resistance to the coffee rust. To confirm the plasmid expression in the plants, the Goose assay will be used since the sequence of the enzyme is present in the plasmid and not in plants. A fluorometric assay will be used because the enzyme can produce a fluorescent product that can be quantified with a fluorometer. To confirm the protein's expression, the Western blot technique will be used after extracting the proteins from the leaves and stems, doing the electrophoresis and identifying them with polyclonal antibodies. In order to make the transitory transformation, we decided to use the binary vector pCambia2301 because it has already been used to make transformations in coffee using a binary system, and because it can be used for a permanent transformation if needed by adding a Werner-Halper plasmid. This vector contains the sequence for the expression of the beta-glucuronidase protein that will be used as a reporter to confirm the transformation of the plants during the lab analysis, and because it contains genes that confer resistance to kanamycin, which will be useful to select the transformed E. coli along with the blue-white screening technique, that will be used to reproduce the modified plasmid for industrial production of the product. We decided to induce the expression of the antifungal proteins, beta-1,3-glucanase and chitinase from the fungus Trichoderma viride, whose proteins have shown a strong effect against phytopathogenic fungi, including the coffee rust. The coding DNA sequence of the proteins was obtained from the NCBI database. The expression cassette of the proteins was introduced in the multiple cloning site of the plasmid, specifically in the restriction site of EcoR1. This site was chosen because the enzyme cuts the plasmid only in this position and because it doesn't cut the parts added to the plasmid, which was confirmed using the GenScript restriction enzyme map analysis tool. The parts introduced in the vector were the CAMV35S promoter, which is a strong constitutive promoter from the cauliflower mosaic virus. Then, a proper Cossack sequence for dicotyledonous plants was added to foster the translation initiation, followed by a sequence that codifies for a signal peptide that will allow the secretion of the proteins of the cells, so that they can attack the fungus in the apoplast. For the beta-1,3-glucanase, a signal peptide from our coffees, beta-1,3-glucanase, was used. The sequence of amino acids of the protein was obtained from the NCBI, and using the tool Signal P 5.0, the signal peptide was identified. In order to get the corresponding DNA sequence for those 28 amino acids, the tool reverse translate from sequence manipulation suite was used by using a specific codon usage table for the coffee tree, which was obtained from the codon usage database. This sequence was substituted for the sequence that corresponded to the signal peptide of the fungal glucanase to have greater levels of protein secretion. After the signal peptide, the sequence that codifies for the fungal beta-1,3-glucanase was added, followed by the nopalin synthase terminator that includes a poly-A signal. Then the procedure was repeated for the expression cassette of the chitinase, using the same promoter, a Cossack sequence, a signal peptide from a coffee's chitinase, the coding sequence of the fungal chitinase, and the same terminator.
In summary, our product is an easy-to-apply spray on coffee crops for farmers to use it in an efficient way. A final label was developed for the product in order to show the application instructions in an easy way.
After experimental data has been collected of the modified E. coli growth, a kinetic growth model can be developed. There are different types of kinetic growth models used to describe a specific bacterial growth rate, where one of the earliest and most used is the monad model. Kinetic growth models are useful because they assist us engineers to design and control biological processes. We propose to use the monad model to obtain the kinetic parameters mu max and K sub S. The following is the monad model. To determine the volume of the reactor, we propose using the following equation to calculate the spacetime. Multiplying tau by the flow rate, we obtain the volume for the reactor. One equation is for a continuous flow stirred reactor, and the other one is for the plug flow reactor. It must be evaluated which one is the better for the process and the experimental data obtained to determine the best reactor to the process. To obtain spacetime for the continuous flow stirred reactor, use this. To obtain spacetime for the plug flow reactor, use this. With respect to biosafety and biopolicies, in Guatemala, if our product idea is to be implemented, certain protocol must be followed. First of all, it should be noted that MAGA, the Ministry of Agriculture, Livestock, and Food of Guatemala, is the institution most related to the synthetic biology in our country. This institution implements the technical regulation on biosafety of living modified organisms for agricultural use, which aims to comply with the Cartagena Protocol on biosafety of the Convention on Biological Diversity. This applies to living modified organisms for agricultural use in transport, research, experimentation, management, development, production, and commercialization activities. To carry out any of these activities, an application must be first be made and approved. If our product is approved by MAGA, we recommend reaching an agreement with this institution and patenting it in the Intellectual Property Registry, so that the product is not used for malicious purposes and its application is responsible and under the supervision of MAGA. On the other hand, among the most important biosafety protocols to be followed when the synthesizing, industrial scaling, and experimenting with our product is the use of the appropriate clothing for the laboratory, as well as the necessary protection, such as laboratory clothes, gloves, glasses, and helmet. We propose restricting people access to the laboratory, as well as following strict cleaning protocols before and after people enter, to prevent any biological parts from escaping from the laboratory. Additionally, experiments should be carried out in the controlled place to prevent the product from spreading in the environment. And once the laboratory work is finished, all the biological parts should be deactivated, using incineration, for example, as well as adequate cleaning of the work areas.
We believe our project has everything it needs to make a sustainable change in agriculture. We want to make sure that our project has the best impact it can possibly have. To ensure this, we have designed a special business plan model to help our product reach more people. Our project is mostly directed to farmers who have had significant losses in their crops due to fungus. It is estimated that around 10 to 15% of crops are lost due to sickness. Fungi is responsible for at least 70 to 80% of these losses. Therefore, these farmers need a fast solution in order to reduce them. The best way to promote our products will be through announcements, whether we do it via national news or signs in rural areas. To make sure that our product is working efficiently, we're going to give samples to farmers affected by fungi and ask them to give us their feedback. Our clients will pick up their products in stores specialized in all sorts of items related to agriculture. After they get their product, they will be able to communicate with us and give us the feedback about it. The key to a successful business plan is publicity, sale, and distribution. All of these aspects have been carefully thought and designed in the most efficient way possible. We have decided to partner with some chemical industries and with a shipping company that ships to rural areas. This way we can make sure that the product is available to more people who might need it. We have also decided to partner with research centers so that we can keep developing the project. With this plan, we're making sure that our project is being advertised in the best way it possibly can.
We try to understand the effects and impacts of coffee roasted breaks to design a profitable alternative for farmers. So we resorted to the International Coffee Organization and the National Coffee Association. We found that coffee represents 1.6% of GDP in our country, generating 500,000 jobs, but in 5 years, roast caused a loss of approximately $543 million and 115,000 jobs. We also had interviews and virtual tours with coffee growers, who told us that their roast reduces production and increases cost. For fighting it, they use easy-to-place, fast, and long-lasting fungicides. But some can't afford the extra cost, so they were forced to cut their crops. After interviews and workshops with the MAGA and fungi experts, the fungicide target was changed from Fusarium to Emileia bastatrix to facilitate spray application on affected plants. On the other hand, the feedback given by the Guatemalan Biotechnology Commission helped us to improve the plasmid design and add 3 methods of verification of gene expression in leaf cells. It was also taken into account that the gene was expressed only temporarily, to avoid damaging local flora and fauna as a secondary effect in the medium term. We also took the suggestion of the synthesis of carbon dots from wasted coffee beans to make a circular economy. At the same time, it was proved that the project has been accepted by the Guatemalan coffee growing community, after a brief explanation of how carbon dots work.
iGEM is a competition that connects us to the rest of the world. To make the most out of this opportunity, we decided to meet with different teams all around the world. Some of the teams we met with were
Chaos, Yucatan, UNAM UNMSM, Aishora, Ojinsimbayo, and Agrobio.
We frequently had meetings where we could get to know them better and spend quality time with them. In these meetings, we shared our projects and exchanged helpful feedback. We also had the opportunity to learn about problems all around the world and proposals to solve them.
To share our experience working on our project and to share more about synthetic biology, we had a variety of activities. We were interviewed on a national news channel called Guatevisión, in which we shared our project and explained the impact it will have on our community. We also created comics about synthetic biology and shared them to make it easier for people to understand what it is. We also shared our project in many schools with the hope to inspire children to be fascinated with synthetic biology and to be interested in learning more about it. Hopefully, in the future, these children will be leading their own projects, inspired by IKTANKAANEK.
Throughout the Design League season and our first participation in iGEM, we learned a lot. We learned the importance of synthetic biology, its beginnings, its applications, and how it can change the world as it can be a profitable and sustainable form of making things. We learned how to make a multidisciplinary team and to work together to accomplish all the objectives of our project. We learned the importance of the design process as the first step in the iGEM engineering cycle to make robust experiments with all the recordings as possible, along with the design of a functional DNA construct that will solve a local and a global problem. We were able to design a methodology that will allow us to make a transitory transformation in coffee plants in order to induce the production of antifungal proteins by the plant itself, which will confer resistance to the coffee roast, Emileia bastatrix, the major pathogen in coffee, by using carbon dots as a transfection method. The carbon dots are planned to be synthesized using wasted coffee grains to promote a circular economy, and they will allow the transformation in planta, so that our product, drizzled biodots, can be applied to already existing crops to increase productivity in the fields. We learn how to communicate our project to stakeholders, our community, and the importance of synthetic biology to other people in order to promote the development of this field in our country. We learn how to include biosafety and biosecurity in our design. We met other teams and exchanged ideas and suggestions. And finally, and most importantly, we had fun during all the competition, and our passion for SynBio was awakened.